Hey everyone, we are going to go over lab 4 and 5 today. This is the first week where you are going to have two separate labs due on the same day. So you want to make sure that you give yourself plenty of time to complete these. This first one can take anywhere between 2 to 3 days because you're growing organisms, and then probably like a total of 3 days to complete. And then your lab 5 can take, same thing, 2 to 3 days for growth, 5 days total to complete. You need to keep the agar plates from this Kirby-Bauer experiment because you will need them for lab 6. So let's go ahead and go into lab 4. You are going to learn about exponential growth. You're going to look at what does viable plate count mean, because if you're counting, you want to be able to see the colonies. If there's too many, then it's going to create problems where you can't identify the microorganism, right? This goes into dilution. There is a mathematical formula that you would have to use to learn about, diluting the sample. Okay, this is showing you how to practice dilution because that's what you're going to be doing in your experiment. So make sure you really review this part really well. And then you've got your experiment. So, right, bleach, distilled water, nothing that you haven't seen before, right? You are going to need to have some measuring spoons in this particular lab. Now, in your lab kit, you need your pipette, you need your nutrient agar, so another nutrient agar tube. You're going to need 3 of these, and you're going to need 6 petri dishes. So you need petri dishes that have already had, agar poured on them. And then another yeast packet. So if your yeast didn't work in, lab 2, make sure you go get that active dry yeast from the grocery store so that it will actually work and your experiment will come out okay. All right, so you're going to need 6 agar plates that have been poured, right? You are going to dilute the yeast. So you're going to need to activate it by putting it in that warm water, let it froth. If it doesn't froth, then it's not good. You need to get another yeast packet. You are going to have 6 plastic cups that you label with 10 to the negative power 1 through 6. Then you're going to do that same labeling on your agar plates. All right, this is going to be your dilutions. So you're going to be careful whenever you're sterilizing your pipette because, right, obviously if you leave it in there too long and it doesn't dry all the way, it can kill your yeast. You're going to put in some distilled water into each of your 6 plastic cups. When you get ready to pipette your yeast, kind of swirl it a little bit, and then you're going to put 0.25 milliliters of that yeast solution into the cup that's labeled 10 to the negative 1. Pipette the solution up and down so that way all the bacteria is rinsed. And then basically do, the same thing with taking the so then you're going to take your 10 to the negative 1 cup and you're going to get 0.25 milliliters out of that cup and put it into the cup labeled 10 to the negative 2. So you're diluting it as you go through. So you're just going to repeat this process all the way till you get to 10 to the negative 6, because the hope is that whenever you get to 10 to the negative 6, it'll be diluted to where you can see individual colonies, right? So then you're going to inoculate your agar plates with the solutions in the cups that you did. So you're going to take 0.125, so about 4 drops from that 10 to the negative 1 cup, and you're going to place it onto the agar plate. Then you're going to put the cover, you're going to swirl it to kind of spread the solution out, And you're just going to repeat that next step, right? So you're going to take, your 10 to the negative 3 cup and put some on that 10 to the negative 3 agar plate, right? Now you can get rid of the cups whenever you're done, and you're going to incubate your agar plates, right, in the same place that you've been incubating all of your materials. You're going to be looking at, how do these colonies develop and what are the plate counts. So typically it would show like, 10 to the negative 1 would have the most, whereas 10 to the negative 6 should have like individual colonies. And so you're going to have to, take photos and kind of record your data. So if we kind of look at that, right, like here's, right, so you're going to say how many are in there and the viability of them. If there's too many, it tells you what to do about that. So you're going to do that for each dilution. Photo of your 10 to the 1 plate, right? Your 10 to the negative 2, negative 3, negative 4, negative 5, negative 6 pictures. And then for data table 2, you are going to be doing, some math here. After that, you do not need these, the plates. You are done. You're going to answer your questions, make sure you do your evaluation questions, and then we're going to go into lab 5. So with lab 5, where did it go? Oh, here it is. Sorry about that. We are going to be testing antibiotics. So you definitely want to make sure if you have any antibiotic allergies that you wear gloves. So when you go to the exploration, it talks to you about different kinds of antibiotics. It'll talk to you about, penicillin versus novibiocin, gentamicin, and then it talks to you about, okay, how do we perform this Kirby-Bauer test? And then you've got questions to answer for your materials. You need that skin sample from lab 2, right? Like you need to make sure that it's still alive and that it's still turbid and growing. If not, then you need to, re-inoculate. All these other things you've seen before. For your lab kit, your antibiotic discs, they should have been placed in the refrigerator as soon as you got them. So take them out of the refrigerator. You need a nutrient agar tube. You need tweezers. You need 2 poured Petri dish plates, and all the rest of these materials. So looking at how to perform this, you need to have the 2 agar plates poured. You're going to take one of those plates and just store it for a future experiment. You are going to divide the agar plate that you poured into, 3 and write down the antibiotics that you're testing in each of the sections. Then you are going to get that skin sample broth, right? Flame the lip, put the swab into, where the bacteria is, transfer the broth, swab it on the agar plate. You want to swab the entire surface of the agar plate, right? So over the whole thing. And then you're going to let it sit until that broth has been absorbed by the agar. Then you're going to get some tweezers. You're going to sterilize them, and then you're going to put an antibiotic disc in the correct section. So you put the novibiocin in the novibiocin section, the penicillin in the penicillin section. You need to be gentle. Like you're going to press it with the tweezers, but you don't want to jam it. Okay. The idea is that it kind of sits on the surface, but it's not going to come off. So you want to let it sit there for about 10 minutes to let those discs attach. Then you're going to incubate it. You're still going to keep your skin sample vial. You know, you're going to disinfect everything, and then you're going to wait. You're going to let it incubate 24 to 48 hours. You're going to look at your plate, and you're kind of looking for, like, if we go back to, right, you're looking for this, like, to happen. So you're, right, like, if you don't have any, like, if there's no zone of inhibition here, like if there's no clear area, then that means that the antibiotic is not effective. Whereas, you know, larger areas means, you know, effectiveness. So kind of going back to, you know, what you're, you know, you've got questions to answer, but if we kind of look at the data table, you have a hypothesis. Basically, you're saying like, okay, I think, you know, whichever antibiotic is, which one you think is going to basically perform the best, and which one you think is going to, you know, the bacteria is going to be resistant to. You're going to have a photo of your plate after you incubate it, right, with your antibiotic discs. So basically your result photo. And then you're basically just going to put in, you know, what is the zone of inhibition measurement. And it tells you how to do that, you know, in here. And then you will go to your evaluation and you will be done.